Good morning, class. Let's go ahead and get started. Let's go ahead and talk about anti-vaxxers today. So welcome everybody to the last mandatory lecture of this class. I hope you guys have enjoyed this class so far. Hopefully you guys have taken away a lot of things that you're going to be able to take with you after this class. And if you ever have any questions, feel free to, reach out afterwards. Before I get into the lecture, I want to remind everybody that I would appreciate it if you guys fill out those evaluations. I haven't checked to see what the status was, but last time I checked, I think we were about 15 or 20%. So if you value the stuff that I've taught in this class or value me as an instructor, I would greatly appreciate any of that feedback that's on there. I'm going to be hosting a review session tomorrow. It's going to be offered during our normal class time. This is optional. You're not going to be required. I'm not going to take any kind of attendance. For this review session, though, I'm going to be taking people's questions. So if you guys have any questions from any of the material, this is the time for you to ask. What I'm mainly going to be going over are things that students seem to struggle with. And I'm using the information based off the midterm and lecture quizzes to see what concepts people were struggling with. I know that there were some people that struggled with the transfer RNA and the anti-codon questions. You will end up getting a couple of those. So I will go ahead and re-explain some of that so you guys have an understanding and a couple of other things. But please, if you have any questions, come to the review session and ask them. There should be no reason for you to do bad on the exam, especially if it's an open note and open book test. I'm not going to be posting the slides for that class, the review session, as some of the questions I'm going to be, that are going to be on there are going to be similar to what I'm going to ask on the exam. And so, it's a good idea for you guys to go to the review session. I will post the recording, though, of the review session only if I get 80% of evaluations in by the end of the review session. So if I do not have 80%, then you guys are tough out of luck. And only the people that came to the review session are going to be the ones that benefit. I'm not going to have office hours on Thursday, as I'm going to count the review session as my office hours, as it's not mandatory. But if you do need to meet with me, feel free to schedule a meeting with me. You have a couple more days before the exam, so there is still time to ask questions. As I said yesterday, the exam is going to be roughly 64, 65 questions. And it's going to be cumulative, where 30% of the material is going to be from before the midterm, and then 70% of the questions are going to be from after the midterm. So, really have an understanding of the material, especially the immunology. So for today's class, I'm going to be giving you a brief intro to the anti-vaxxer movement. I'm going to talk about some of the obstacles we've encountered when it comes to vaccination. I'm going to talk about science and its relation to media. I will talk about fake news and false information and how that can be detrimental to our understanding of things and confusion. And then I'm going to give you one key reliable source that scientists use when it comes to trying to find reliable data. So for this class, I want you to understand why anti-vaxxers have a fear of vaccines, but also understand that anti-vaxxers have been around for a long time. Know that this fear and the spread of false information, especially in the context of vaccines, has detrimental consequences. Understand that media tends to oversimplify our scientific findings. I want you to know the tools that you can use to detect online bullshit. I want you to be able to critically think about the information that is given to you. I feel like in this country in general, we passively take in information and we don't really critically think about it. And I'm hoping that some of the material that I've shown you in this class, you guys are actually thinking about instead of just And there is some memorization, it's science, you're learning terms. But, more importantly, understand how the whole concepts work, how they're all interconnected with one another. I want you to know the difference between a primary and secondary source, as this is pretty important when it comes to trying to distinguish between reliable data. And then understand why peer-reviewed journals are considered reliable. So anti-vaxxers. So anti-vaxxers are an old issue. They have been around since really the development of the first vaccine by Edward Jenner. So here's a political cartoon of Edward Jenner giving a vaccine to somebody, and then the people that have already been vaccinated are turning into cows, as this vaccine was meant for smallpox, but the vaccine was derived from cowpox, as we discussed in previous lectures. So here is a brief timeline of anti-vaxxers. So we have the beginning of the discovery of the vaccine against smallpox in 1796. And then sometime later, you basically have groups that are forming against vaccination, and majority of it is out of fear. And so here we have a league that formed out of the UK, and then a short time after that, there was a league formed in the United States. Eventually, there's more leagues that were formed, as you can see in the early 1900s, and these people are going to spread false information, even though science was still developing, and it's still developing now, but we didn't know a whole lot back in the day. Now we know a lot more than what we did. And so it's kind of a shame that as time has progressed and we've developed new vaccines and new therapies to fight against diseases, you have these people that are basically telling people do not get vaccinated. And a lot of their claims as to why people should not get vaccinated are unfounded. And so the fear of vaccines has a couple of different reasons. So, one, I think, is people always fear what they don't know. But two, there's been a couple of incidences throughout history that manufacturers have actually messed up the development of the vaccine. And once that vaccine is given to a person, it ends up leading to some kind of detrimental disease. And I'll highlight some of those road bumps in a couple of slides. But the one that I'm really going to emphasize is this disgraced scientist, Andrew Wakefield, who basically falsified data and said that the MMR vaccine is linked to autism, which we now know is not true. With that information being published, and on top of that, you have public figures basically advocating for anti-vaccination, you have this whole counterculture movement that began saying, stop vaccinating your children. And in the early 2000s, we basically had almost a full eradication of polio and measles in the whole world. But as these counterculture anti-vaxxer movements built up popularity, we started to see less people having their children vaccinated. And then with that lack of vaccination ended up leading to a lot of different outbreaks in different places in the United States and in Europe. And so it's a shame we have this kind of technology to be able to prevent these diseases, but people think that they know better than doctors and scientists, so they choose not to use the technology out of fear and misinformation, and then we have epidemics. So one thing that anti-vaxxers love to use is pseudoscience or fake science. And here I have a couple of lists of examples of people that use pseudoscience. So you have people that want to use essential oils and stuff to cure whatever disease you have. Ridiculous. You have the anti-vaxxers, which I'm highlighting in this lecture. You have the people that don't believe that climate change exists despite all of the data that exists out there. And then you have people that don't believe AIDS or COVID are an actual thing, despite many people succumbing to these kinds of infections. And so when we look at the difference and compare the difference between science and fake science, we see some of these key principles. We see that, with scientists, they adhere to a well-recognized scientific method. We observe things, we come up with a testable hypothesis, something that we can actually, design experiments for to get data out of. Once we generate those, the experimental design, we can actually perform the experiment, so we can go ahead and get information out of that. Once we have the information, we can go ahead and analyze it and draw conclusions from it. Well, with pseudoscience, there really isn't a process. They find random things online that usually don't have a legitimate source, and they use it as facts. And when they do that, that is their way of basically rationalizing their ideas for why they believe whatever they believe in is true, and whatever scientists say is fake. When it comes to science, you want to be able to reproduce the results. And that's why a lot of scientists, you'll see in papers, they not only have multiple replicates, but they also have 2 or 3 independent experiments. And the whole point of this is to make sure that the data that you're getting is reproducible and thus reliable. Now, if you're getting a lot of variation between your samples and repeated experiments, then more than likely, whatever you're trying to test probably isn't a thing. When it comes to pseudoscience, you typically can't replicate whatever experiment they're claiming that they performed or no. It's usually somebody's opinion. Oh, I heard it from somebody, or I saw it on somewhere, I read it from somewhere, but the people that they're hearing or seeing this from aren't reputable sources. When it comes to science, some of the claims can be testable, and if they are reproducible and show what scientists have done before, then you can validate whatever it is that they're trying to test. Now, if people repeat those experiments and they get a different result, or they test it in a different way that maybe is more reliable, you can disprove certain ideas. Whereas in pseudoscience, it's all based off of beliefs. You think of something, but there is no logical sense behind how you test it. Typically with science, you have somebody reviewing whatever it is that you're doing. Whereas pseudoscience, usually, you can get the facts, but a lot of time they will refute the facts because they know better than scientists and doctors. And then lastly, when it comes to good science, there are multiple different people that are showing the same thing. And that's generally how the things that we learn in science when it comes to what's in our textbooks is multiple people have shown the same thing. And if they've shown the same thing in a reliable way, then it becomes a fact. Now, with pseudoscience, they typically will, if they do end up taking some kind of data, it's usually a small amount of their sample size, and small sample sizes cannot be extrapolated to the rest of the population, which I will highlight in a couple of slides. A lot of pseudoscience people or anti-vaxxers will typically look for these kinds of experiments that were performed to fit whatever they're trying to claim. And a lot of the time when you look at the literature for the data that they're trying to cite, the experimental design is just way too small to be able to make any true conclusions. So, when it comes to the anti-vaxxers, there is a sense of fear, and there is a sense of legitimate fear, and it has to do with manufacturers basically messing up in the process. And I'm going to highlight three different incidences that this occurred. So when we were trying to eradicate polio in the 60s and 70s, what ended up happening was you had this company called Cutter Labs that didn't actually quality control check their polio vaccines. So usually vaccines will be made in different batches, and then typically people will take a couple of different samples from those and test to see their efficacy. Well, with this lab, they did not do any of that quality control. And so with the polio vaccine, if you remember in the vaccine lecture, one of the ways that this vaccine works is they have an inactivated form of the virus. And they typically will do this by adding some kind of fixative such as formalin. And so formalin will basically kill the virus, but keep its structure intact so the body can recognize it. Well, with this company, they didn't add enough formalin to the virus, and so the virus was still alive. And so when they ended up giving this to children, basically they ended up getting infected. And this resulted in 250 children getting polio. And if we remember with how polio affects us, it basically affects our nervous system. And so a typical textbook image of a child that has polio will end up developing something called muscular atrophy, because their muscles are no longer being stimulated because they're no longer walking, and they typically are going to have very reduced muscle mass. The other incident that I want to talk about is the development of RotaShield, which was a company that was trying to make a vaccine against the rotavirus. And so the rotavirus is a type of virus that typically infects the intestinal epithelial that will end up causing diarrhea and a bunch of other things. And so we were trying to be, trying to create a novel technique. And so we engineered the live rotavirus as a vaccine in order to basically educate our immune system so we could have memory against it. But there were a couple of issues when it came to the development of this vaccine. One, they didn't really do something called a titration titer, where you basically want to figure out what is an optimal amount of virus that you can give a person that is going to generate an immune response, but without getting them sick. But what was found with this particular vaccine was there was just too much of the virus within each dose. And so with the multiple doses and a high amount of virus that was present, it just wasn't good for the body. And what they ended up finding was this intussusception, which is just the intestinal folding. And once they discovered that this was occurring in the intestine, they basically pulled it off the market, and the vaccine just was stopped. They stopped the development of this. And so this is another reason why there's fear of vaccines, because we don't know, or there's a fear of what we don't know is going to happen to our body. And then the last one I want to highlight is the 1976 swine flu. So in 1976, there was an outbreak of swine flu in the United States. And it caused a lot of concern because when you compare the genetics of the swine flu and the Spanish flu, they were very similar. And so if they were very similar, then that meant there's this possibility of it becoming a pandemic. And so in order to fight against it, the Ford administration basically decided to create a national swine flu immunization program. However, this program was very rushed. There wasn't proper quality control check, and there were no safeguards put into place. And on top of that, we always think that these politicians have our best interests, but they don't. It was an election year, so he was just trying to do this to get reelected. What ended up happening, though, because this vaccine was rushed and the proper controls were not put into place, they ended up finding an association with this particular vaccine with Guillain-Barré syndrome. And so GBS is a type of paralytic disease. And so usually it happens when the virus ends up getting into your neurons. So it could be either, in your brain or your motor in the periphery. But what ends up happening is because your body is recognizing that there's an infection present, your CD8 T cells come and start to kill those neurons. And when you kill those neurons, then that's going to lead to paralysis. And so for this reason, after they discovered that this vaccine was causing this, the vaccination efforts were halted. And this is one of the more, I guess, claims that a lot of anti-vaxxers will end up using when it comes to citing the fears of why you shouldn't get vaccinated. So when we look, though, at the overall types of vaccines that have been developed and that have been given us since we were babies, they have been involved in getting rid of a lot of diseases and preventing a lot of childhood death. We were able to eliminate smallpox because of the vaccine campaign that was done in the 60s and 70s. But more importantly, in a lot of these developing countries, you have reduced childhood death because of these vaccines. So we really take this for granted in this country, especially the anti-vaxxers. But because of popular press and different kinds of counterculture groups, there has been a resurgence of some of these preventable diseases because they decided to not listen to scientists and basically used unscientific data. And so one of the main people we have to thank for really, adding fuel to the fire of the anti-vaxxers is Dr. Andrew Wakefield. So this is the guy that is responsible for saying that vaccines cause autism. And this paper has been discredited. There have been multiple people that have looked at it, and the data was basically falsified. So here is a picture of disgraced Andrew Wakefield. I don't even consider him a scientist. Scientists do not falsify data, and if the data is what it is. And so this basically happened in 1998. Him and his research team found a link between autism and the MMR vaccine, which is supposed to prevent measles, mumps, and rubella. Basically, when this idea became popularized, and we have Jenny McCartney, who went on to the Oprah Winfrey Show, that really helped further cause this cascade of misinformation. Now we have this counterculture movement. And so this controversy not only caused a lot of controversy within the population, but it's still going on till today. When good scientists reanalyze the data that his paper came out with, we learned that the data that was in there was all falsified. And we now know part of that had to do with he was being bribed. There were lawyers who were fighting court cases for parents that had given their children vaccines and they ended up developing autism. And so to help strengthen these lawyers' cases against the vaccine producing companies, a lot of the lawyers cited his work. And so if you have scientific data that has been published, then that can be used in the court of law to help be used as evidence. But after all of this analysis, we now know that all of it was fake, and his paper has been disproven. And if you look online for the paper, it's going to say retracted right on it. And that means that it's been disproven and whatever is in it is fake. And it wasn't until, and this didn't occur until 2010, which is like 10 years after the fact. So you had 10 years of anti-vaxxers using this for their rationale for why they don't want to vaccinate their children. And unfortunately, the effects of this are still rippling through time now. Now, when we look at the consequences of these lies, we see the changes of people getting vaccinated and the amount of people that are developing these kinds of preventable diseases. So here in the mid 90s, you had the majority of people getting vaccinated. And then when his work came out in 1998, we started to see this decrease of people getting this particular vaccine. But, unsurprisingly, with this reduction of people that end up developing or that aren't getting vaccinated, you're going to see a rise in the confirmed cases of measles. And so this data is generated in England, but we have similar trends that have happened here. And so notice how when we look at the amount of cases, they're pretty low all throughout up until, I would say, the early 2000s. And, there's a variety of reasons for this, but one of them has to do with this concept of herd immunity. And I had you guys watch a video for one of your labs. Basically, once enough people are vaccinated, even though there's some people that are unvaccinated, because you're breaking that chain in the spread of the virus, you're able to basically protect people that are unvaccinated. When you have a reduced amount of people that aren't vaccinated, then you're going to have less of that protection, and there's going to be less chains that can be broken. And so when that occurs, then you're going to end up seeing rises in cases that could have easily been preventable. Here we have the omnibus autism proceedings. So basically, this was a set of court cases that examined the claims of autism vaccines causing autism. And so you had people after the publication of Andrew Wakefield's work, basically using this to get compensated for their child developing autism. And so basically in these court hearings, there were two main arguments that were used. There was one that both the MMR vaccine with vaccines that contain isomerisol were causing autism. And after many studies of being performed by credible scientists, we see that there is no causational factor or effect of vaccines and the link to autism. And then the second theory is, after they disproved the MMR, then they were thinking, okay, it's the mercury in the vaccines that's causing it. And this is not the case. Again, scientists, good scientists, credible scientists have disproven this. And then we also have to consider the form. So if you haven't noticed, in all of my class, I'm always talking about the structure of things. The structures are so important when it comes to how our body processes things. And so when we look at the structure of thiomesanol, we see that mercury is part of this compound that's in a salt form. And the whole point of this molecule is to preserve the structure of inactivated viral proteins or particles. Because it has this structure, we are able to excrete this. We absorb it, obviously, but we're able to process it and excrete it. It does not bioaccumulate, meaning it does not build up in our bodies. If you were just taking mercury by itself, the component Hg, then yes, you would end up absorbing that, and that would lead to toxicity and a bunch of other brain issues. But it's in this form that can be easily excreted by our bodies. Mercury that's within this molecule that is used to preserve the vaccines does not cause autism, nor does it bioaccumulate. So the other main culture issues we have when it comes to vaccinations are people that basically are continuing to spread false information. So while vaccines are not prohibited by Muslim law and are actually promoted by major Muslim medical associations that people will trust, there's people that still claim that Western vaccines do a variety of things. They are a CIA plot to cause AIDS and infertility. They have things in them that Muslims aren't able to consume because it is against Muslim law. And then you have things saying that there's harmful substances that are going to destroy our immune system. And, I feel like I have to give some credit with this fear when it comes to referring to the CIA. So I've told you in the past, I love history. I'm a scientist by training, but I like to see how humans have developed with over time because we always repeat. If we look at a lot of the things that happened in this last century, especially in Latin America, our government, especially in the CIA, did a lot of bad things in Latin America. And that's why we've had a lot of influx of immigrants from Latin America in the last 50 to 60 years. So you can go ahead and do your own research on that. But with those illegal activities that they've done, and some of them are actually sanctioned by Congress, there's a legitimate reason why some other countries, some non-Western countries, have this fear of vaccines. But because of these claims, we have an increased resistance to people wanting to get vaccinated in some of these African and Asian countries. And because of this, we're seeing a resurgence of polio. And so when you look at this graph, don't just passively, take this, okay, there's a lot of virus that's happening in Africa and in Pakistan and in Afghanistan. When you look at the actual graph, you see that wild polio, so polio that's just naturally found in the environment, that's typically found in Asia. Now, unfortunately, some of the vaccines that are being given in Africa are also causing polio. And I don't know if scientists know why that is. I can only hypothesize that there's a lot of people that they don't have enough nutrients in their diet. And I've already shown you lectures on how important nutrients is when it comes to the health of our bodies, but also the health of our immune system. And so my thought is, if you have people that have malnutrition and aren't able to give their bodies or the immune system in particular the right things to help generate that memory response, then they're not going to be able to fight off the virus. And so a lot of the polio vaccines have a weakened version of the virus. And so if they have a weakened version of the virus, but they also don't have good nutrients that are going to support the immune system, the virus can potentially win. And so I think that might be a rationale as to why that's going on, but I really have no idea. So let's look at science and the media. So are media and science really at odds? Well, it really depends on your perspective on things. When we look at the media and when it takes scientific data or scientific findings, they usually just are very direct when it comes to whatever it is that they read. Most of the time, the people that are reading this are journalists that have no scientific background whatsoever. When they give these results, it's very simple and very unambiguous, making it seem like it is very fact, which a lot of times science is not a fact. As I was explaining earlier, you need multiple groups of different scientists showing the same thing for it to become a fact. The media is all about catching the consumer's attention. And so that's why they usually try to keep it short and sweet so they can capture the reader, so they can read or listen to whatever it is that they're trying to say. In reality, though, when it comes to science advancements, it's very incremental. We find discoveries, but those discoveries aren't really considered facts until a lot of people have shown that. A lot of the time, science is not unambiguous. And this has to do with a lot of science is hypothesis driven, meaning that we observe stuff, and then we make an educated guess on what we think is happening. And then we test that educated guess, that's our hypothesis. And then a lot of the time, they're wrong. A lot of times scientists are wrong. We think something is, we take in the information and data that's out there, try to rationalize it with logic, and then we test it. And based off the findings, you're either going to have positive or negative results. Both are going to generate data and both are going to tell you something. But unfortunately, when it comes to publishing work, a lot of journals like positive data, things that are going to show some kind of change. But it's also important to know things that don't have a change, because that's also going to be leading to wasted time for other scientists. A lot of the time when it comes to scientific findings, it's usually going to be interested to scientists that are within that field. So, for example, I personally don't really know anything about ecology. I know, the basics of it, but when it comes to reading any of those kind of papers and stuff, I'm not interested in it. I like reading scientific journals or papers that are going to help me understand the immune system and some of the current therapies that we're able to use because of these different findings that we've been able to do in the last few years. So here I have a nice diagram that kind of simplifies the difference between scientists and journalists that are presenting media. When it comes to doing scientific work and publishing that data, it takes a lot of time. It takes months to years. However, a journalist can just find a bunch of things that they find online and then, make some conclusions and then write that up. When it comes to the area of expertise, the years of a scientist being at the bench and doing this work is going to make them an expert. And they're going to be able to do the work, get it published, and be able to write whatever their findings are. When it comes to journalists, they're writing very fast. They're not, really taking in all of the true information that is present. And I feel like if you're a scientific journalist, then it's a good idea to either have a scientific background or at least educate yourself on the scientific terms. And I'm hoping if there's any communication or journalist majors in the audience, I'm hoping that this class has shed light on some of this terminology, because it's very important when it comes to being able to comprehend scientific data. And I'm going to highlight some of that in a few slides. Usually when we're publishing data or for a paper, usually we're going to have the heading of the article is going to be kind of long, because it's going to take in consideration the main finding and then if there's a protein or certain kind of mechanism that's involved, and if there's a specific kind of species that involved. Whereas journalists or people that are involved in media will oversimplify what is actually learned, and instead of understanding all of what's here, they basically just associate one gene that is like, so for example, this particular thing is saying that in their mouse studies, there was a gene that was associated with developing a certain kind of blood cancer. But there are certain types of mice that have these receptors or proteins that where those numbers change drastically. Whereas the journalists will end up extrapolating this and oversimplifying it onto the general population, that this gene is involved in cancer, which it may or may not be, but you have to take into consideration the species that it was done. We do mouse experiments because mice, when you compare our genes to mice, they're 70 to 80% genetically similar to us. But not everything that we learn from mice can be extrapolated and put onto the human population. That's why there's clinical studies that are done to validate some of that information. So let's go ahead and take an example of this. So we have the D614 gene mutation in the scientific literature. So back in the middle of last year, there was this COVID variant that basically had a point mutation where this amino acid was changed to a glycine. And so in this paper, I have a quick excerpt of this paper. And basically what it's saying is that there's this change in this amino acid. This is where a genetic variant ends up coming. And there is a link between its transmissibility, and that's based off of the data that they're able to generate from their model and with what's out there in the human population. But an area that I want to highlight is that in here it says that patients that are infected with this particular variant seem to have higher amounts of virus, basically in their nasopharynx in the initial infection. But there is very little evidence that there's a significant relationship between the kind of virus that is, or whatever the genome of the virus is, and how harmful the virus can be. They stress that the linkage between how severe the virus can harm us is based off of our medical conditions and our own genetic factors, not the virus itself. Now, and if you read some of this, it might sound a little confusing, and so that's why it's important to have people that have a science background to be able to interpret this. Now, let's see what the media said. The media oversimplified this and said coronavirus mutations emerge and that this may bypass mask wearing and hand washing protection. So this is saying that the virus can basically mutate itself to where it can pass through masks, and hand washing is going to have no effect on it. That's fucking ridiculous. If you read the article, it says nothing about hand washing or mask wearing. It just talks about this point mutation that occurs within the body, and it can potentially lead to higher amounts of virus, but the virus itself is not having an effect on the severity of disease or being able to bypass these barriers. It's ridiculous. And so when you actually read the paper and highlight some of the key things, when usually in the end of the paper, the discussion section, the authors will, they'll say there's a possibility. There's always a possibility that these point mutations can lead to an advantage of the virus. And there's times where it can, but in general, it doesn't. The mutations actually make the virus less harmful to us, because again, viruses do not want to kill us. They need us to live. They need a host cell to live. If we're dead, the virus dies. And so in this short paragraph in the paper, they basically say there is the possibility that this amino acid change can increase the spread. But the reason why we're seeing an increased amount of this variant within the population has probably more to do with our social behavior. During the Memorial Day weekend, there was more regulations that were more relaxed. People were able to gather more, and a lot of people weren't wearing their masks. So if you don't have those barriers, then of course the virus is going to be able to spread. Yes, it has that point mutation, but I don't think it's the point mutation itself that's causing increased transmissibility. But that's something that can be tested. So now let's look at this other drug that we all heard about, hydrochloroquine. And so this was promoted by the French physician and microbiologist, Dr. Raoult. And so this is the name of his paper. He basically said this drug and azithromycin, an antibiotic, can potentially be used as a treatment for COVID. And so again, the media likes to over extrapolate what's in there. But when it comes to what you're seeing in the media, always look at what the source is and then critically think about what the source is saying. And so what did the actual data say? And so feel free to go ahead and read this. But basically, the main critique that I feel like this paper has is the sample size is way too small. You have 6 patients that were asymptomatic, 22 people that had upper respiratory infections, and 8 people that had lower respiratory infections. This sample size does not represent the rest of the world. On top of that, you have to consider in these studies is not only is the sample size big enough, but is it diverse enough? Are there different kinds of race involved? Are there different kinds of sexes involved? Are there a variety of ages that are involved? If the answer is no, then you cannot extrapolate this data onto the rest of the human population. On top of that, they're using an antibiotic to fight off a viral infection. And again, I've emphasized this multiple times. This is going to lead to bacterial resistance. And so when the media basically got a hold of this, they ended up saying that this drug combination could shorten coronavirus. And they say it as a matter of fact. And it is never a matter of fact. I'm going to say this over and over again. Multiple scientists need to be able to confirm stuff in order to consider it to be a fact. And then we had our Dr. Trump going ahead and backing this up, saying that it can actually work. And people listen to him. Obviously, we've seen all the problems when he existed or when he was in power, and a lot of people are going to listen to him. Now, when we actually look at scientists that perform future studies or further studies on this, when they increase the amount of patients that were involved in their studies, it was shown that there was no link between these 2 drugs being able to help with COVID. Somebody said a huge problem during COVID was that everything became political, especially, yes, for sure. And unfortunately, a lot of politicians do not have a science background. We need to listen to the scientists, and that's one of my biggest issues that I've had in the last couple of years is when you call scientists into question, especially the credible scientists like Dr. Fauci, who have been, doing viral research for decades, who actually know how the whole system works. And then you have people like different kinds of politicians that are calling his integrity into questions like, no, your integrity, politician, is in question. So now let's talk about fake news and false information. So, we've all heard about fake news, and fake news is basically news or information that's trying to be passed off as fact by a reliable source. But you really need to be skeptical of everything that you hear and read. You want to really consider who is telling you this. Are they actually credible? How do they know? Where do they get their sources from? Are those sources reliable? What are they trying to sell me? Usually there's some kind of ulterior motive that they have behind that. So let's go ahead and take this poster example. So in this ad, they're basically trying to tell people that the virus is a hoax and it's not as deadly. And the reason for that is as of November 16, 2020, that there's only been 2.5 million people that have died overall from all causative deaths. So this includes COVID, heart disease, cancer, everything. And so with these numbers in play, you can compare it to previous years where we see that there's this increase of deaths per year, but then in 2020, we have this sudden drop. And so if we have this sudden drop, then they're trying to make the reader think that COVID is not as deadly as we make it believe. Well, you should always question, not only the numbers, but where are they getting it from? So, always try to find the source. And so when they look at the source, we see that, okay, this number actually matches. It's all causes of death is 2.5 million people in 2020 as of November 16th. But you also want to question the numbers. How are those numbers being generated? And the best thing you really want to look at and consider is, when were those numbers taken? And so if you look at the dates of when this number was taken, you see that it's from February to November. This number does not account for any time in January, nor does it take in consideration the last 6 weeks of the year, which tend to have higher rates of the flu, which tend to lead to death in a lot of the older patients. So if we just use a little bit of critical thinking, we can go ahead and estimate how many deaths should be expected in 2020. And so if we take the amount of deaths that are present here, divide that by the amount of weeks that this number falls into, we see that this equates to almost 60,000 deaths per week. And so if we multiply that by the number of weeks that were not accounted in the original figure, this comes up to almost 600,000. And so if you add this 600,000 with the number that's already present, we see that reaches around 3 million deaths. And then when you actually look at the actual numbers, the deaths of last year were 3.3 million. So always question the source. And once you found the source, if it's a credible source such as CDC, then question the numbers that are being presented. And so obviously this poster is bullshit. Here I have a couple of ways to help you be able to decipher bullshit that is found online. What you always want to look at is, do multiple sources corroborate the finding? Are there multiple people or groups, reliable groups, that are showing the same thing? You want to be skeptical of the source. If it is a suspicious source, and a suspicious source typically is going to have something that has no citations or links to it, then it's going to typically not be something that's reliable. If it does have links attached to it, go to the primary literature. Make sure that it's actually coming from a reputable source. And I will give you a link to what a reputable source is, and I'll highlight that in the last part of the lecture. Yeah, it's very important when it comes to reading the scientific literature that's out there. There is reverse images sites that you're basically able to find the sources of where certain images come from. And I highly recommend looking at this site where it gives you a little bit more about what deepfakes are and how some of the software is able to be generated. I'm sure some of the computer scientists in here are aware of this. And I went on the site and I tried distinguishing what was fake and what was real, and I had trouble. So, be aware of that. And then I have a couple of lists of fact-checking websites that you can use if you want to see if something is real or not. So there is some time left, and so I wanted to show you this video that was sent to me by my conspiracy theory of an uncle. And so give me 1 second, and so it's like false information like this that just makes me really angry because people will believe it. So here it goes.
Okay, we're live on We Are Vax. Vax to Instagram. Right, people, we have a bit of information. I want you to listen, hear this incredibly well. This is a fact. I want you to share this everywhere. I want you to screen grab it. I want you to copy it, whatever it takes, because they will take this down. This is a fact. Share to all of your Bible groups or anybody else that doesn't want aborted fetal tissue fragments put into them or their DNA changed. This is a fact. This is the packaging of the AstraZeneca COVID-19 vaccine. Okay, right in front of you. I want you to look at this. Okay, I want you to look at it in lists, ingredients, blah blah. I'm not interested in that right now. I want you to home in on this here. This is the COVID-19 vaccine. Claire's just going to pull this up for you. This number, okay, it's called CHAD, whatever that is, 0 or whatever it is, times 1. Okay, this is the CHAD 0 times 1, the COVID-19 vaccine. So Claire, go to the next one. Just wait 1 second, guys. Okay, so Then you're going to go to researchsquare.com. It's really important, people. The reason we're doing these videos is because I want you to learn to do your own research. Don't rely on us or anybody else. Do it yourself. So we look up this CHAD 0x1 COVID-19. And the first thing, before we go to what I've written there, is I'm looking and I'm thinking, oh, what's recombinant? What does that even mean? So I asked the new and I asked them, everyone's like, gosh, don't know. So this is what you need to do. You need to Google every single word on here. Don't skip by it thinking, oh, that's science, don't understand. So Claire, let's look up this word on Wikipedia, and straight away, there you see it. Recombinant, it's DNA. Our DNA molecules formed by laboratory methods of genetic recombination, such as molecular cloning, to bring together genetic material from multiple sources, creating sequences that would not otherwise be found in the genome. Okay, you do that research yourself. But my point is, again, it's right in front of your face that this stuff is in this vaccine. So they can call us crazies and terrible hosts that spread terrible fear-mongering things. They're putting it right in front of your face. Okay, go back to that thing again. So what we're asking you to do is do your own research. Okay, we've started it off for you. So back to this number. That means that AstraZeneca package I just showed you, this is their vaccine. And as they go down, again, we use direct RNA sequencing to analyze transcript expression from the CHAD, let's call it the CHAD vaccine, genome in human MRC5. So you think to yourself, oh, let's skip past that, a little bit complicated, must be a bit scientific. No. Look it up. And what do we find, Claire, when we look that up? MRC5 is originally developed from research deriving lung tissue of a 14-week-old aborted Caucasian male fetus. There you have it, peeps. This COVID-19 vaccine that everybody is saying is going to save the world has a truckload of shit in it, but one thing it definitely has is the lung tissue of a 14-week-old aborted Caucasian male fetus.
It's been replicated over and over and over again for a long time.
So you send this to every single Christian-loving pro-life person. Forget.
All right, that's enough out of you. So, okay. A couple of things with this. I'm going to start for the disclaimer. This vaccine does not have lung tissue in them. I feel like videos like this have been spread throughout everywhere, and people are fearful of the vaccine because of this. My mother-in-law is afraid of that because she doesn't support, abortion and stuff like that. Okay, so a couple of things. So yeah, sure. The paper that she's referencing is a credible one, but science is a language. If I were to try to read some kind of foreign language and try to look up different words, I'm still not going to be able to comprehend what it's saying. Science is the same way. You need to have a true understanding of what these words are in order to generate a full comprehension of what it's saying. And I'm hoping that this class has shed some light on some of that terminology for you. So I know that at times it might have been a little bit difficult for the material, but again, it's for your own benefit. I only have the best interest for the students. And so what the paper is saying is it tested that vaccine in lung tissue, lung cells. So the A549 and that was it MCR7 or whatever it was, those are two cell lines that people will culture in petri dishes. It's called in vitro culturing. And typically that's going to be the first thing that scientists will do when they're testing things before they put it into an animal or human. And so what it's saying is that it used those cell lines because they express the ACE2 receptor. They were trying to see the efficacy of how it worked. So what that lady was saying was she was interpreting that whatever that paper was saying was what makes up the vaccine. That is not how it works. We've gone through the lectures already on how the vaccines work, and there were no signs of lung fetal tissue in them. All right. So now it's time to give you a reliable source of that all scientists use. And this place is called PubMed, and I have a link here for you. And so if you're ever reading some kind of article that has information in it and they have citations, check to see if that paper that's cited is within this database. If it is not within this database, then you cannot trust it. This database is basically going to be full of different kinds of scientific articles. And the kinds that you're going to find are mainly going to be primary and secondary source articles. So the primary source articles are going to be how we generate the data. So what that lady was looking at, that is a primary source, but she didn't understand what it said. The primary source is going to be how we test different things, different hypotheses, and generate data and draw some conclusions from that. Whereas a secondary source, such as review articles that are found on PubMed, are going to typically, whatever their topic is of interest, say like they're trying to look at efficacies of different COVID vaccines, they're going to go into the primary literature sources, read on all these different sources, and basically do a summary on them and look at the different efficacies of them and how they're generated and whatnot. If they're in here, they're going to be reliable sources to use. So if you ever are interested in looking up a certain specific scientific topic, go to this site and look up whatever it is that you want. A lot of the time, whatever you're interested in is going to be found in there. Now, the reason why this site is so reliable is because of the publishing that it goes through. Every article that gets published is peer reviewed. And basically, this is going to be by people that are able to evaluate the work because they're experts in the field. They're people that are qualified and actually know what is going on in whatever field that you're applying or trying to publish your manuscript in, and they will end up critiquing it. And they're going to end up sending their critiques to the editor, who's in charge of if it gets published or not. And based off of whatever the experts say, there's going to be a couple of different routes this goes. They're going to just reject it altogether. They're going to say it doesn't fit whatever we're trying to publish. They will send it back and tell you, okay, most of it's good, but you need to revise a couple of figures, or you need to do a couple more experiments to support your hypothesis or whatever you're trying to show. And very rarely on the first time, your manuscript will get accepted where it's completely flawless and it's good to go. And so when you're looking at reliable sources, here is a checkmark list of things that you should consider. You want to look to see if the information's current. The information is constantly changing. If you remember at the beginning of the pandemic, Dr. Fauci kept on changing the different kinds of protective measures or mandates because of the information and data that was being generated. We know a whole lot more about the virus now than we did a year ago. You want to consider if the data or whatever the source is a primary or secondary source, which I've already highlighted the differences between those two. You want to see who wrote those articles, who wrote those sources. Are they somebody that you can easily find online, and are they well qualified? Are they known in their fields? Does the author lack any kind of conflict, a conflict of interest? So if there's some kind of, a lot of time it's a monetary conflict of interest, then they're going to skew the data so it fits whatever they are trying to sell. So if you look at a lot of studies that are done by food companies, usually the data is going to be skewed in a way that supports whatever they're trying to sell. So be conscious of that. In that particular paper, are there the data or the information that they're giving, are they also cited? everything is always going to have some kind of source for it where the information comes from. Are the experiments that are within the primary source described well enough for other scientists to be able to reproduce? Reproducibility is very important when it comes to reliable data. You want to consider if the information has already been peer reviewed by experts. And then is the information unbiased? So again, usually that goes along with conflict of interest. If you have some kind of motive, usually you're going to have some bias when you're doing your work. And then what is the information trying to tell me? Is it valid or is it not? And they're just trying to make a bunch of false claims. All right, so there is a bit of time. And so this next thing isn't so much science related. It's more of, it's a video that I saw a few years ago that I feel like if you guys haven't seen it, hopefully it'll spark some interest and maybe compel you to want to change the system that we have today, because we have a very broken system. And I've highlighted a couple of different systems, such as the healthcare system in the food industry. And you guys are going to be the future leaders of tomorrow. You guys all have a variety of different backgrounds, and you guys are getting educated for a purpose. Utilize that education to lift up your communities. That's what we need. And if we realize that the issues that we have in our society are more of a socioeconomic one rather than a race one, then we would be able to unify a lot better. So
there's a chart I saw recently that I can't get out of my head.
You guys to see.
A Harvard business professor and economist asked more than 5,000 Americans how they thought wealth was distributed in the United States. This is what they said they thought it was. Dividing the country into 5 rough groups of the top, bottom, and middle 20% groups, they asked people how they thought the wealth in this country was divided. Then he asked them what they thought was the ideal distribution. And 92%, that's at least 9 out of 10 of them, said it should be more like this. In other words, more equitable than they think it is. Now, that fact is telling, admittedly, the notion that most Americans know that the system is already skewed unfairly. But what's most interesting to me is the reality compared to our perception. The ideal is as far removed from our perception of reality as the actual distribution is from what we think exists in this country. So ignore the ideal for a moment. Here's what we think it is again. And here is the actual distribution, shockingly skewed. Not only do the bottom 20% and the next 20%, the bottom 40% of Americans barely have any of the wealth. I mean, it's hard to even see them on the chart, but the top 1% has more of the country's wealth than 9 out of 10 Americans believe the entire top 20% should have. Mind-blowing. But let's look at it another way, because I find this chart kind of difficult to wrap my head around. Instead, let's reduce the 311 million Americans to just a representative 100 people. Make it simple. Here they are: teachers, coaches, firefighters, construction workers, engineers, doctors, lawyers, some investment bankers, a CEO, maybe a celebrity or two. Now let's line them up according to their wealth. Poorest people on the left, wealthiest on the right, just a steady row of folks based on their net worth. We'll color code them like we did before based on which 20% quintile they fall into. Now let's reduce the total wealth of the United States, which was roughly $54 trillion in 2009, to this symbolic pile of cash. And let's distribute it among our 100 Americans. Well, here's socialism, all the wealth of the country distributed equally. We all know that won't work. We need to encourage people to work and work hard to achieve that good old American dream and keep our country moving forward. So here's that ideal we asked everyone about, something like this curve. This isn't too bad. We've got some incentive, as the wealthiest folks are now about 10 to 20 times better off than the poorest Americans. But hey, even the poor folks aren't actually poor, since the poverty line has stayed almost entirely off the chart. We have a super healthy middle class with a smooth transition into wealth. And yes, Republicans and Democrats alike chose this curve. 9 out of 10 people, 92%, said this was a nice, ideal distribution of America's wealth. But let's move on. This is what people think America's wealth distribution actually looks like. Not as equitable, clearly, but for me, even this still looks pretty great. Yes, the poorest 20 to 30% are starting to suffer quite a lot compared to the ideal, and the middle class is certainly struggling more than they were, while the rich and wealthy are making roughly 100 times that of the poorest Americans and about 10 times that of the still healthy middle class. Sadly, this isn't even close to the reality. Here is the actual distribution of wealth in America. The poorest Americans don't even register. They're down to pocket change, and the middle class is barely distinguishable from the poor. In fact, even the rich between the top 10 and 20 percentile are worse off. Only the top 10% are better off. And how much better off? So much better off that the top 2 to 5% are actually off the chart at this scale. And the top 1%, this guy, well, his stack of money stretches 10 times higher than we can show. Here's his stack of cash, restacked, all by itself. This is the top 1% we've been hearing so much about. So much green in his pockets that I have to give him a whole new column of his own because he won't fit on my chart. 1% of America has 40% of all the nation's wealth. The bottom 80%, 8 out of every 10 people, or 80 out of these 100, only has 7% between them. And this has only gotten worse in the last 20 to 30 years. While the richest 1% take home almost a quarter of the national income today, in 1976, they took home only 9%, meaning their share of income has nearly tripled in the last 30 years. The top 1% own half the country's stocks, bonds, and mutual funds. The bottom 50% of Americans own only 0.5% of these investments, which means they aren't investing. They're just scraping by. I'm sure many of these wealthy people have worked very hard for their money. But do you really believe that the CEO is working 380 times harder than his average employee? Not his lowest paid employee, not the janitor, but the average earner in his company. The average worker needs to work more than a month to earn what the CEO makes in 1 hour. We certainly don't have to go all the way to socialism to find something that is fair for hardworking Americans. We don't even have to achieve what most of us consider might be ideal. All we need to do is wake up and realize that the reality in this country is not at all what we think it is.
At PNC Bank, we believe in the power.
Well, that's my lecture, everybody. I hope you guys enjoyed this class. Again, tomorrow will be the review session. And if you have any questions, feel free to email me. And if not, I will see you guys during the exam.